Good afternoon, students. No one has joined it. No? Yes, good afternoon all. Good afternoon. I hope everyone preparation is going well. TK. Yes, good afternoon. TK. Today we are going to see previous year question paper for 2024. TK. ICAR PG 2024. TK. Before going to start today's section, first I want to tell some of the important thing. TK. This 2024 question was little much tricky. TK. Little much tricky and it's bit conceptual also. So pay your 100% effort. TK. Pay your 100%. TK. So we can discuss all the things properly. TK. Kindly try to what interact in the section also. TK. TK. We are going to see first 60 questions in this session. TK. First question. Which of the following molecule can be classified under glycosphingolipid category? Okay. Which of the following molecule can be classified under glycosphingolipid? Okay. Any guesses? Glycosphingolipid. Yes. Glycosphingolipid. Yes. Excellent. Sulfamide. Okay. We know the glycosphingolipid will have ceramide backbone. It will have a ceramide backbone. Here sugar will be attached. Okay. Here fatty acid will be attached. Okay. So cerebroside means what? Cerebroside means the ceramide having one molecule of glucose. This is clear. Ceramide backbone having a molecule of glucose is known as cerebrosides. TK. You have to remember here, TK, if this cerebrosides will have a sulfate group, that we will say as sulfatide. TK, sulfatide. So the right option for glycosphingolipids. Sphingolipid having glycose. That is known as what? Sulfatide. TK, clear? Yes. Next, the bicarbonate buffer. Bicarbonate buffer is a principal buffering system in blood plasma. This we already know. They are asking the ratio of bicarbonate to carbonic acid required to maintain the pH at 7.4. You have to observe here. TK. See, they are asking what will be the ratio of bicarbonate to carbonic acid. Okay, bicarbonate to carbonic acid required to maintain the pH of 7.4. Okay. So from this question they have given pH is 7.4. Okay, pH they have given 7.4. Okay. Then concentration of bicarbonate to carbonic acid we have to find. Okay. So a equation that is correlating the pH, pKa and the concentration of acid and base we have already seen here. Henderson-Hasselbalch equation. Okay, Henderson-Hasselbalch equation we can use to solve this question. Okay, we can use to solve this question. What is Henderson-Hasselbalch equation? pH equal to pKa plus log bicarbonate concentration by H2CO3 minus concentration. I hope this is clear for everyone. Okay. Now, pH value they have given 7.4. We know the pKa for this carbonic acid is what? Bicarbonate buffer 6.1. This is very important thing. pKa is 6.1 plus we have to find the concentration of this thing. Okay, just a simple mathematic thing. 7.4 minus 6.1 equal to log concentration of bicarbonate by H2CO3 minus. Okay. So if you minus this means what it will come? It will come 1.3. 1.3 equal to log concentration of bicarbonic acid by this thing. Okay. Now, how to solve this one? Okay. Just take antilog. We have to take antilog. If you take the antilog, we can know the concentration of bicarbonate. Okay, concentration of bicarbonate to carbonic acid is 10 power 1 power 10 power 1.3. Okay, 10 power 1.3. If you further simplify this, the value will come around 19.95 approximately. So the ratio required is what? 20. From approximately we are getting 19.95. So you see here, so we need a ratio of 20 is to 1. 20 is to 1 to maintain the required pH of 7.4. This is very important question. Okay, and you have to observe the we have to apply the Henderson-Hasselbalch equation to know the relationship of pH, pK and concentration of its acid and base pairs. Okay, clear? This is very important. Okay. Next question, they have asked from beta oxidation in plants. Okay, beta oxidation in plants. Observe the assertion, the disruption of beta oxidation of fatty acid. Disruption of beta oxidation in fatty acid in plants can potentially reduce the stress-induced ROS accumulation. They are saying if beta oxidation was disturbed, that may reduce the accumulation of ROS in plant under stress condition. Yes, this is true because in plant the beta oxidation primarily occurs in where? Peroxisome. Okay. In peroxisome, what it will happen? The fatty acid will be there. By the action of fatty acid oxidase, this will produce hydrogen peroxide in spite of FADH2. So this may be lead to oxidative stress. So this is correct statement. Assertion is an correct. Reason what they are saying, in plant, beta oxidation of fatty acid takes place predominantly in peroxisome. This we already see in our classes. Peroxisome is the major site for beta oxidation, where first enzyme for beta oxidation is fatty acyl-CoA oxidase that will produce large amount of hydrogen peroxide. So assertion and reason is both statement are correct and reason is correct explanatory for assertion. Okay. So both A and R are correct and R is the correct explanation of A. This is clear, students? Clear? Like this, they have asked more conceptual question. You have to be attentive in the class. Okay. Next, what they are asking, the serum level of which of the following metabolite can be efficiently distinguished. That means if vitamin B12 deficiency is there, yes, vitamin B12 deficiency is there, which component will be accumulated more in the blood? Yes, many can come. Excellent. Methylmalonyl CoA. This we have already seen in our fatty acid catabolism class. Fatty acid catabolism class we have seen. Okay. If an odd carbon fatty acid will undergoes to beta oxidation, at final 3 carbon compound propionic acid will form. This we have already seen. For the conversion of this propionic acid to succinyl CoA, different enzymatic reactions will come. Okay, different enzymatic reaction will come. Among them, methylmalonyl CoA mutase. This is the major enzyme that will catalyze the last step for conversion of the succinyl CoA. The most important thing, the cofactor for this enzyme is vitamin B12. Okay, vitamin B12. So, if this cofactor is not there, this methylmalonyl CoA will be accumulated in the cell. By that, we can easily differentiate there is an vitamin B12 deficiency. Clear? Next, they have given the different enzymes and what are the classes of enzyme. This we have already seen in our enzymology class. Pyruvate carboxylase. Any guesses, student? What will be the answer? Pyruvate carboxylase. What class it will come? Fumarase, phospholipase and nitrogenase. What classes it will come? Any guess? Yes, any guesses? This we have already discussed. Yes. Any guesses? Digestive enzymes. Digestive enzyme will always comes under the classes of hydrolytic enzyme. Okay, hydrolytic enzyme. From this option they have given phospholipase. Okay, phospholipase will always come under hydrolase enzyme class. So C will have option 2. See here where C will having option 2. Only this will have option 2. So this will be the right option. Correct. Pyruvate carboxylase, what it will do? Pyruvate, it will bind with carbon dioxide to form oxaloacetate. Here CO2, it is attaching with this pyruvate to form oxaloacetate. So that is a ligase reaction where ATP also utilized. So A will come option 4. So this correct. Fumarase. Fumarase is what? It's an lyase enzyme. This we already seen in our lecture itself. Fumarase is an lyase enzyme. Then nitrogenase. Here NADH will be act as a source of reducing equivalent. It will comes under the class of oxidoreductase. So the right option for this question is option B. I hope this is clear for everyone. Okay. Here another thing, they have given match question only. They have given different sequencing techniques, like sequencing. What can we do? Sanger sequencing, like the different protein sequencing techniques they have given, and what is the major things, what is the major chemistry of that things they have asked. Okay, you see here, among these we are very familiar with Sanger sequencing. Sanger degradation, what it will do? Sanger degradation, what it will do means what? From Sanger degradation, we can determine the N terminal of the amino acid. This is clear for everyone. Yes. Yes. N terminal of amino acid we can determine. Okay. How N terminal we are determining? We are using fluorodinitrobenzene. Okay. This will go and bind with the N terminal. So N terminal we can determine by using this Sanger sequencing. Okay. Then hydrazenolysis. This is specific for C-terminal determination. Okay. So A will come option 1. Okay. D will come option 4. So this is the right option. Okay. Then alkaline or acid hydrolysis. This is very simple question. You know. Then trypsin digestion. This also we have seen in our basic classes. Trypsin, what it will do? It will cleaves the bond between lysine and arginine. This we have already seen. It will cleaves the bond between lysine and arginine. So we can say site specific cleavage of polypeptide chain will be there. Yes, we have solved this one. Next, another important thing, they have asked about linkages of beta glucans in different organisms. Okay, observe the question properly. What they are asking: chemical linkage between beta D glucopyranose residue. Okay, in the structure of beta glucans. Okay, beta glucans, they are asking if that beta glucans was identified from different sources like cereals, mushroom, bacteria and yeast. What are the different combinations will be there? Okay, question is clear. So they are asking about beta glucans. What will be the linkage of beta glucans in these different species? Okay, we know, yes, we know A, B and D. Okay. Yeah, they are saying option A. Okay. So we know the plant will have beta glucans, like wheat and barley will have beta glucans. That will have what linkage? Beta 1,3 and beta 1,4 linkage will be there in plants. Clear. But bacteria also will have the same linkage. Mushroom also will have same linkage. But the speciality of yeast is it will have beta 1,3 as well as beta 1,6 linkage. Beta 1,3 as well as beta 1,6 linkage. You see here beta 1,3 is common to everything, but beta 1,6 or beta 1,4 will be there. There will be a never existence of beta 1,4 along with beta 1,6. So option D is ultimately wrong. Okay, so wherever D is coming, that is ultimately wrong. Okay, so what will be the answer? A, B and C will be the right option. I hope this is clear for everyone. Okay, another question. These things we have already discussed in our lecture itself. They are asking about inhibitor from glycolysis pathway. We have specifically seen this one. Iodoacetate, where it will go and bind? It will go and bind the enzyme glyceraldehyde 3-phosphate dehydrogenase. This we have already seen. It's a glycolytic enzyme. This is true only. Yes, we have seen. But iodoacetate is a chelating agent. No, it is not a chelating agent. That chelates the magnesium ion. This is not a chelating agent. Yes. So the right option is what? A is true, but R is false. What is the exact mechanism of this iodoacetate? What it will do? The enzyme will have an cysteine amino acid. it will have a SH group. Okay. This inhibitor will go and bind here. Because of that, active site of the enzyme will be inhibited. So the enzyme catalysis will be stopped. I hope this is clear. So statement 1 is correct and R is false. Okay. Next question from TCA cycle. See, metabolic questions they have asked so much metabolism question. Here what they are asking about TCA cycle, they have given the question. Which of the following statement is true about citrate? They are asking about citrate. So citrate, what it will do? It's a 6-carbon compound. This we know. Okay. This will be formed by acetyl CoA and oxaloacetate condensation. Okay, you see here whether in option is there. Yes, it is formed by Claisen condensation reaction. So this is correct. Yes, wherever C is coming, that is correct. So our bad luck, they have given C in all the option. Okay. Then second thing, the most important thing about citrate is it's a prochiral carbon. Okay, it's a prochiral carbon containing compound. Okay, this is a specific for citrate. Okay, so this option will come. So it is not a chiral carbon, it's a prochiral carbon. It is not a chiral carbon. So C and D is correct. Where C and D is coming? C and D is coming here. Yes, only here only C and D is coming. So this is the right option. Why it is not a secondary alcohol? Here comes the question. Why it is not a secondary alcohol? You see here, if you see the hydroxy group attached in the citrate molecule, it will be bonded to carbon atom that will be bonded to 3 different carbon atoms. Clear? That's why we are saying this is a tertiary. It's a tertiary alcohol. Okay, it's not a secondary alcohol. It's a tertiary alcohol. So the right option for this is C and D only. Yes, they have answered correct option only. B is the right option. TKB is the right option. Another most important thing from fatty acid, they are asking question. The basics only, they have covered so much of basics as well as conceptual question. Okay. Anyone can solve this one? What? Dihomo gamma linolenic acid. Dihomo gamma linolenic acid. Okay. Is whether it is omega 6 fatty acid or omega 3 fatty acid. Okay. You have to know how it has been formed. Okay. They are asking how this di-gamma linolenic acid is formed. Before going to that things, I will just give some brief introductions. Okay. We know alpha linolenic acid. We everyone knows this will have a linkage what? 18 carbon will be there, 3 double bond will be there. This is correct. 9, 12 and 15th position double bond will be there. This is an omega 3 fatty acid. Omega 3 fatty acid. This we already know. Then we have to know about gamma linolenic acid. Gamma linolenic acid. The specialty of gamma linolenic acid is it is also having 18 carbon and 3 double bond, but the position of the double bond will be there at 6, 9 and 12. This is clear. So this will be what? It's an omega 6 fatty acid. I hope this is clear. This is an omega 6 fatty acid. Next, another important thing, in our question they have asked dihomo gamma linolenic acid. Okay, simple. Dihomo gamma linolenic acid means this gamma linolenic acid will enter into an one cycle of elongation. Elongation means what? One molecule of acetyl CoA will come here. Okay, so 2 carbon will be added. I am right. So 20 carbon will come. There is no action of desaturation. So the double bond will have same number of double bond. Okay. Then the position of the double bond will be shifted. Position of the double bond will be shifted. So it will come from 4. Okay. Like this, the position of the double bond will shift. Okay. 4, 7 and 9. Clear? So the position of double bond will be shifted. Now we are coming to the question. What they are asking? Dihomo gamma linolenic acid. They have given 8, 11, 14 will be there. Double bond they have given. Okay. It's an omega 6 fatty acid. In our last slide we have seen this will have 20 carbon. Then double bond is ending at 14. So this is an omega 6 fatty acid only. So this is an right option and it's an omega-3 fatty acid. It's correct. Then second thing, formed by conjugation of 2 linolenic acid. This is wrong. This is just forming by elongation reaction. Formed by elongation of gamma linolenic acid. Yes, this is correct. Formed by isomerization of gamma alpha linolenic acid. This is also wrong. So what will be the right answer? A and C. So option C is the right answer. Okay, you have to apply the concept. This is very important point. You have to apply the concepts. Okay. Another important things. See, from technique in biochemistry, they have asked all problem questions only. Okay, all mathematical questions only they asked. Listen the question properly. This we have discussed in last year question paper, 2025 question paper itself, they have asked like this. Okay. In spectrophotometry, if transmittance value of a solution is 75%, they have given. Okay. Then its absorbance would be equal to what? They are asking what will be the absorbance, like that they are asking. If you remember in yesterday class, we have seen absorbance equal to 2 minus log transmittance percentage. This we have seen in our last class. Yes, money can come. Excellent. Option A is the right answer. We can see the solve. We will solve this now. Okay. So 2 minus log percent is what? 75%. Log 75 is what? 1.875. So what will be the right option? 0.125. Clear? See, in 2025, they asked the formula. In 2024, they asked the problem. So, repeated question is there here. You have to see here, repeated question is there here. Okay. Next question. In dual affinity nitrate transporter, if a transporter, nitrate transporter is dual affinity, okay, how both a low and high affinity transporter is regulated? Okay, in which condition it will be high affinity and which condition it's an low affinity? How this regulation is happening? Okay, so dual affinity means what? It will behave both as high affinity transporter or low affinity transporter. Which modification will regulate this high affinity to low affinity? This is very important thing. Okay, there will be a particular threonine residue. This will be phosphorylated by kinase enzyme. Okay, this will regulate this affinity of this transporter. I hope this is clear. So specific phosphorylation, that means phosphorylation state will regulate this affinity. Okay, whether it's a low affinity or high affinity. Okay. Next, this is another simple question. They have asked from enzymology, what are the enzymes and how it is regulated? What are the post-translation modification happening and how these enzymes are regulation? Okay, by allosteric activation and regulation, they are asking. We know among this most important thing, we are very familiar with nitrate reductase, NR. This will be regulated by what? This is very clear, by phosphorylation and 14133 protein binding. It will link. It will regulate by phosphorylation and 1433 protein binding. So for option C, one should be there. Option C, here only one option have option C. This is clear. So only one option have option C. Then second thing, you see nitrogenase. Nitrogenase enzyme is typically, yes, it's a typically regulated by what? By ADP ribosylation. This is another important question. So by ADP ribosylation, this nitrogenase enzyme will be regulated. Then glutamine synthetase, that will be regulated by adenylation. Okay. Rubisco will be regulated by S nitrosylation. Okay. So we have to choose the option by elimination method also. Okay. Clear? Next, pyruvate carboxylase catalyze the conversion of pyruvate to oxaloacetate is a which reaction? Any guesses? Whether it's an anaplerotic reaction or cataplerotic, elimination or hydrolytic reaction. What will be the answer, students? Yes, any guesses? It's an anaplerotic. Yes, excellent. It's an anaplerotic reaction. Okay, it's an anaplerotic reaction. Why? Because the intermediate of TCA cycle can be siphoned off for some of the biosynthetic process. the gap will be filled by anapleuritic reaction. So, this pyruvate carboxylase, this pyruvate will bind with carbon dioxide, that will lead to the formation of oxaloacetate. This will go and enter the TCA cycle to fill that gap. Okay. So, this is a type of anapleuritic reaction. Okay. Then amyloids. After hearing this word itself, you have to know amyloids means what? These are misfolded proteins. This is very clear. We know amyloids are what? It's a misfolded proteins. What it will do? It will cause Alzheimer disease or Parkinson disease. It will lead, see, if protein is not folded properly, that are known as misfolded proteins. If these misfolded proteins are accumulated in the body, that may lead to Alzheimer disease or Parkinson disease. This is direct question. Okay, you have to know about the basics. Okay. Next, they have asked about bacteria. Okay, peptidoglycan they have asked. Okay. Assertion, what they are saying, peptidoglycan containing the bacterial cell wall is resistant to hydrolytic enzyme action of many proteases. They are saying the peptidoglycan is there that will have in the bacterial cell wall that are resistant to hydrolytic activation of many proteases, like that they are saying. Okay. What is the reason means? This peptidoglycan will have D amino acids. Okay, it will have a D amino acids. Okay, that may resist the most protease, like that they are saying. What will be the right option? Whether the both statement are correct. Whether if it's correct means whether the assertion and reason are correct. Any guesses? Yes, option A is correct. Yeah. So both the statements are correct. Yes. Bacteria will have peptidoglycan, that will have D amino acid, that will be resistant to protease. Yes, excellent. Excellent. Next, this is another important question. You have to work smartly here, use your brain properly. See, if you see the option itself, we can easily filter the question. Okay? We can easily solve the answer if you see the option itself. See, what they are asking, they have given different forms of vitamin D. Okay? Different forms of vitamin D. Ergo calciferol, cholecalciferol, calcidiol, calcitriol. Okay? Calcidiol and calcidiol. Okay. You see here, ergo calciferol. This we already seen in our cholesterol metabolism itself. Okay. From squalene 2,3 epoxide. Okay. These compounds will be formed like lanosterol will form, ergosterol will form. Okay. Ergosterol. Yeah. Ergosterol will be what? It's an vitamin D2. This will present primarily in what? This will be primarily present in fungi. So first will come what? Option A. Where it's coming? First everywhere it coming option A only. Then second thing, cholecalciferol. Cholecalciferol is what? It is the precursor for other vitamin D synthesis. Okay. So only one hydroxy group will be there. Okay. Cholecalciferol will have only one hydroxy group that will be present at third position. So vitamin D3 is cholecalciferol. Okay. Calcidiol will have what? Two hydroxy group, one at third position and another at 25th position. So C will come this option. Calcitriol, it will have three hydroxy group, one at third position, another at first position and another at 25th position. So what will be the right answer? 1, 3, 4 and so what will be the right option, students? We have solved this. Just let me know what will be the right option for this. What will be the right option? C will come option 3 only. Any guesses? Yes, C will come option 3. D will come. What will be the answer? What will be the answer? B. Yes, yes. So B will be the right option. Okay. You see here, first option is what? For ergo calciferol, vitamin D2, cholecalciferol, what it will do? Vitamin D3. Calcidiol is 25 hydroxy vitamin D3 and calcitriol is 125 dihydroxy vitamin B3. One person asked Ganesh Mahi, he asked which biotechnology book preferred, sir. You can use HS Chawla, okay, Principles of Plant Biotechnology by HS Chawla, okay. That is very standard book, okay, that you can use. You have very less time means just to go and read instant notes on plant biotechnology, okay. These books are good, okay. I am writing here. You can see here first introduction to plant biotechnology. Plant Biotechnology by HS Chawla. This is one of the standard book. Okay. Second thing, instant notes. Instant notes for plant biotechnology. Okay. These two are very standard, very much standard books. Okay. If someone have BD Singh book, that is also preferable only. Okay. BD Singh also. Okay. Next question. Given below are two statement, assertion and reason they have given. Assertion is what? During evolution, some species including humans become dependent on dietary vitamin C. Vitamin C is what? Essential. Why means because of the biosynthetic gene or biosynthetic enzyme was lacking in human. What is the enzyme that is lacking? They have given L-gulonolactone oxidase. Whether it is correct? L-gluconoxidase enzyme is absent in humans. That way, vitamin C is essential for humans. Like that, they are saying whether these both statements are correct and whether the reason is correct. Any guesses? Yes, any guesses? Yes, this is correct. Both A and R are correct, and R is the correct explanation of A because humans lack this enzyme gluconolactone oxidase. If this enzyme is not present, that will not, okay, yes, if that enzyme is not there, vitamin C synthesis will not be there. Because of that, what it will happen? Vitamin C become essential for humans. Okay. So both A and R are true and R is the correct explanation for A. Okay. R is the correct explanation for A. If you want to work out so much of objectives, okay, our mock test series is there. Just you go on by, like that is 499 only. Okay. Those who want to prepare for next year ICAR JRF examination, they can join our course. Okay. That will be available online. Okay. You can join that course also. Okay. Next question. They have asked about Rubisco activase. Rubisco activase. Okay. They have asked about Rubisco activase. We know Rubisco activase, what it will do? It is a ATP dependent reaction that will activates Rubisco for fixing the CO2. This is clear. So it is a ATP dependent reaction. Yes, that is true. In the option D, there is B option is not there. Okay. Second thing, it activates the Rubisco by promoting the entry of sugar phosphate to its active site. Whether it will come? It will activates by promoting the entry of sugar phosphate. No, this will not come. How it will come? Because why? Rubisco enzyme will be there. First carboxylation will happen. After that only this RUBP will come and bind. This is the substrate sugar phosphate. It will come and bind there. So C option will not come. TK. So the right option is A, B and D. I hope this is, yes, Udalakshmi, correct. Option A is the right option. Next, about the enzyme they are asking. What is the other name of GOGAT enzyme? This I have told clearly in the class. You have to answer. What is the other name for the glutamine oxoglutarate aminotransferase, GOGAT enzyme? Anyone can explain? Yes, excellent. Glutamine B. Read the question properly. B. Whether it is a glutamine synthetase? Observe the question. Glutamine oxoglutarate aminotransferase. Yes, C will be the right option. Okay. You observe here in ammonia metabolism. Okay. What it will happen? First, by nitrate metabolism, what will happen? Ammonia will form. Okay. We are applying nitrogen fertilizer that will convert it into nitrate. Nitrate will convert into ammonia. Okay. This is nitrate assimilation. Okay. This ammonia will be assimilated to amino acid. By which pathway? GS go get pathway. This we have already seen in our class. GS means what? Glutamine synthetase. Okay, it's a glutamine synthetase enzyme. Okay. Go get means what? It's a glutamate synthase. It's a glutamate synthase, or we can also say as glutamine oxoglutarate aminotransferase. Okay, so the right option is what? Glutamate synthase. I hope this is clear. This is the purpose of discussing previous year question paper. You have to read the question properly, then you have to select the options also properly. Okay, don't do mistakes. Okay, so the other name of GOGAT is glutamate synthase. Okay, which of the following molecule can be considered as a lipid? Any guesses? Which of the following molecule can be considered as lipid? Ubiquinone, retinol, dolicol, and beta-carotene. What will be the answer? Yes, ubiquinol, retinol, dolicol, and beta-carotene. What will be the answer, students? Yes, this is very simple question. Ubiquinone is a lipid. Yes, we know. Retinol, vitamin A, fat soluble. So that will also a lipid. Dolicol for membrane anchoring, it will be there. That is also a lipid. Beta-carotene. Isoprene units will combine to form this beta-carotene. So this will also be an what? Lipid compound. So A, B, C and D. Clear? Clear? Yes, clear. Yeah, we are going to next question. Okay. The sequence of action of enzymes for fatty acid synthase complex. They are asking, yes, many option C is correct. Now we are going to see next question. The sequence of enzymes that are involved in fatty acid synthase. Okay, fatty acid synthase complex. If fatty acid synthesis is happening, what is the sequence of this enzyme will happen? Okay, we have already discussed this thing. We have already discussed. First acetyl CoA will be there. If you say means acetyl ACP will be there. This will bind with malonyl CoA. Correct. Malonyl CoA will be there. Malonyl ACP will be there. So acetyl CoA is a 2-carbon compound. Malonyl ACP is a 3-carbon compound. This will bind by ketoacyl ACP synthase. So first enzyme is what? Ketoacyl ACP synthase enzyme will come. Then next step what it will happen? It will utilize NADPH. So ketoacyl ACP reductase will come. Clear? Ketoacyl ACP reductase will come. Okay. So you see the option. First will come ketoacyl ACP synthase. So first will come B option. This is the right option. Okay. First will come option B, beta-acyl ACP synthase. Then it will come beta-acyl ACP reductase. Then hydroxyacyl ACP dehydratase. At last, enoyl ACP. Yes, enoyl ACP reductase will come. Yes, excellent. Option B is the right option. Excellent. Okay. At last, thioesterase will come. Thioesterase. This will cleave the fatty acid from the enzyme complex. Okay. This will cleave the fatty acid. Okay. Thioesterase enzyme will come at last. Okay. Clear? Next, assertion and reason. They are giving sorghum is a C3 plant. Yes, this is a wrong statement. Is a C4 plant. Okay. Sorghum, the primary CO2 acceptor is 3-carbon molecule phosphoenol pyruvate. Yes. Any guesses? Sorghum is a C4 plant. So assertion is wrong. So in sorghum, the primary CO2 acceptor is a 3-carbon molecule phosphoenol pyruvate. They are asking phosphoenol pyruvate. Yes. In C4 pathway, the first carbon acceptor is what? Phosphoenol pyruvate. It's a 3-carbon. Yes. So assertion is false, but reason is true. Excellent. Next, they asked about acrylamide. What this acrylamide will do? Okay. This acrylamide will form by Maillard reaction. Okay. You just remember it will form by Maillard reaction. Okay. This how it will form means if we are long time we are cooking or roasting we are doing means that time aspargine amino acid will be there. Okay, aspargine amino acid will be there in the proteins. This will bind with the reducing sugar. Okay, that will lead to the formation of acrylamide. Okay, this is one of the neurotoxin compound. Okay, so this is the simple concept. If asparagine is there, that will bind with reducing sugar by Maillard reaction, that will lead to the formation of acrylamide. Yes, so the right option is what? Asparagine. They are asking it's a toxic compound, carcinogenic compound also. How it is forming? Okay, because the protein will react with the reducing sugar, that will lead to the synthesis of this acrylamide. This is another important thing. Sucrose is what? It's a non-reducing sugar. Okay, so this will not come. Okay, clear? Next, the order of elution. See, another technique in biochemistry question. They are asking so much of applied thing from for biochemistry. Ticket. Particularly in technique in biochemistry, they ask so much question. That to applied aspect they have asked. Okay. See here, the order of elution for amino acid. They have given different amino acid. Whatever they have given, histidine they have given, glutamate they have given, then lysine they have given. They are from cation exchange column. They are saying here they are using cation exchange column. That means what? This column will have negative charge. This can bind the positive charge ions. This is clear. That is cation exchange. With a buffer having buffer pH, they have given 6.0. So the pH of the buffer is 6.0. That will come for all the things. Okay. Then they have given the pKa. pKa for each and every amino acid they have given. pKa for first amino acid is 6.0, second is 4.25, and third is 10.5. We already discussed these things in yesterday lecture itself. They have asked like this question. If pH is greater than Pi, if pH is greater than Pi, that molecule will have positive charge. If pH is less than Pi, okay, if the pH is less than Pi, that will have negative charge. This is clear. So you see here, if positive charge is there, means that will go and bind with the column very tightly. So it will go and bind with the column very tightly. If negative charge is there, means what? It will elute later. Okay, so the order of elution what? Correct. Option A is the correct. Glutamic acid, histidine and lysine. Yes, excellent. Okay, clear. You have to apply the concept here. Okay, another question. They have asked about nitrogen metabolism. See, in this year, they have asked so much of question from technique in biochemistry, photosynthesis. Okay, then plant metabolism, they have asked so much question. Then nitrogen metabolism, they are focusing much. Okay, this we have already discussed. What are the genes that are controlling the nitrogen fixation genes? Okay, we know there are two genes are there, NIF-A and NIF-L. Okay, this NIF-A will act as an activator. It an activator for other nitrogen fixing genes. NIF-L is what? It's an inhibitor of other nitrogen fixing genes. This we have already seen in our class. So they are asking which product or which pair of the NIF genes control the expression of other NIF genes. So what will be the right answer? Yes, NIF-A and L. NIF-A and L is the right option because NIF-A is the activator and L will be the inhibitor for other nitrogen fixing genes. Okay. Another thing, they have asked about DNA supercoiling. Question related to DNA supercoiling. Okay. The DNA of microorganism living under extreme temperature will have predominantly have whether it will have positive charge and negative, that whether it will have negatively supercoiled or positively supercoiled DNA, or it will have relaxed or extensively methylated DNA will be there, like that they are asking. First we have to know what is meant by positive supercoiling, what is meant by negative supercoiling. We know that DNA will have what? Base pairs will be there, that will be there here. Okay. If in per helical turn, if per helical turn, we know 10.5 base pair will be there. This is clear for everyone. Approximately we can say 11 base pair, 10.5 to 11 base pair will be there. If this DNA is accommodating more amount of base per helical turn, that we will say what? It is a positive supercoil. Okay, it's a positive supercoil. That means it will have more amount of bases. Okay, comparing to normal one. If it is accommodating less number of bases, that we will say as negative supercoiling. So, if negative supercoiling is there, means the DNA will be easy accessible for transcription, replication for all the process. Clear? So, stability also will be optimum. But if positive supercoil is there, means the base pairs will arranged more compacted. Okay, more compacted. So, what it will happen? It will increase the TM also. Okay, thermal stability also will be provided because of this positive supercoiling. So, the right option is what? If under extreme temperature, means the DNA of the microorganism will have positively supercoiled DNA. I hope this is clear for everyone. Okay, so it will have positively supercoiled DNA. Next, they have given the different forms of DNA. Then we have to match. See, work properly. We know this Y1 DNA. This is a triplex DNA. This we know. So D will come option 1. Where is coming D option 1? So this is the right option. Okay. D is coming option 1 in this one only. We will discuss remaining things also. A DNA, what it will be there? It will be a short, broad and right-handed. This is clear for everyone. Yeah, this will be short, broader. BDNA means it's a longer, thinner and right-handed. Yes. ZDNA, what it will do is ZDNA, it will have alternate purine and pyrimidine residue. Okay. Then another important thing, this is what? Left-handed helix will be there. Okay, left-handed. So the right option for this is what? Option C. Other question they have asked about mitochondrial respiration inhibitors. They are connecting the mitochondrial inhibitor respiration. You see here, fishermen in certain parts of the world follow a practice of beating the roots of tree. Roots of tree. What roots of the tree? Which root tree they are beating? Okay. In river banks in a manner that transfer the extract of the root. There will be some extract in the root. Okay. This practice paralyze the fish and make them as a easy prey. The most acceptable reason is what? They are saying they are taking some plant root. They are what? They are beating on the bark trees. In a manner, some of the chemical will be released from the root. This chemical is doing something in the fish. Because of that, that fish is paralyzed and that can be easily catched by the fisherman. What will be the exact mechanism? Like that they are asking. Okay. Heavy metal, sure it will not come. Dopamine, not it will not. This will also will not come. The root, yes, option A is correct. Yeah, the root will have a rotinone. They will use dairies plant. Okay, dairies plant roots they will use. That root will have a rotinone. Okay, this will go and inhibit the electron transport chain. Electron transport chain at complex 1. Okay, so there will be a less production of energy. Because of that, the fish will become paralyzed. Then it will become easy prey. Fisherman can easily catch that one. I hope this is clear for everyone. Okay. Next, another thing, basics. They have focused more on basics also. See here, they have given different molecules like alpha keratin, collagen, myoglobin, and concanavalin. Then what are the structures predominant in there? Secondary structure. Okay, whether it will have antiparallel beta sheets, alpha helical structures, or it will be predominant cysteine amino acid like that. Okay, alpha keratin, it will be present majorly in hairs. We know hairs will have more amount of keratins will be there. Okay, this hair straightening we are doing the hair straightening. We are applying an heat on it. Because of the heat, that hair will become straight. Correct? Why? Because the cysteine amino acid that will form a bond with other things. Okay, this will be degraded at high heat or destabilized. Because of that, that hair is become straight. So alpha keratin means what? It will have rich in cysteine residue. So option A will have 2. See, this is the only option it's there. Option A will have 2. Yes, option B is correct. Yeah. Collagen. Collagen we know. Collagen is predominant in glycine, proline and hydroxyproline. And most important thing, Vitamin C is required for cross-linking of this collagen molecule. Okay, this is another important question. Okay, myoglobin, it is predominantly of alpha helix structure will be there. 8 number of alpha helix structure will be there. Concavalin, this we have seen in our last class also. 2025 also we have seen. This can be used for purification of what? Carbohydrates we can use. This is a lectin molecule. Okay, okay. This is a lectin molecule that will have predominance of beta pleated sheets. Okay, beta pleated sheets will be there. So the right option is option A. Okay. Any doubts so far? Any doubts, students? Just solve this question. Which of the following cannot be the form of plant tissue culture method? What will be the right option? Which of the following cannot be the form of plant tissue culture method? Yes, D. Excellent. D is what? Is grape cultivation. It's a not a tissue culture method. Okay. So callus culture we can do, suspension culture we can do, organic organ culture also we can do. So the right option is viticulture. Okay. Another important thing from tissue culture they have asked. Which of the following will be present in highest amount in plant tissue culture? Simple question. Highest amount, what will be there? H3BO4. Here, boron will be there. Okay, this is a mineral nutrient. Ammonium nitrate, N source will be there. Na is an hormone. Glycine is an amino acid. Now you have to tell which will be more. Yes, excellent. Ammonium nitrate will be more because that will act as an exclusive nitrogen source. Okay. Yes. Next, clonal propagation should not be used for multiplication of which of the following species? Yes, this is another simple question from plant tissue culture. I already told in our lecture itself, plant tissue culture, they will ask question that will be simple only. Okay. What will be the answer? Just apply your mind. Okay. Slow growing culture, this can use. Yes, clonal propagation we can do. Wheat seedling, we can use. Vegetative propagated crops, we can use. But we can use for male sterile lines because the male sterile lines will be maintained by what? Male sterile lines will be maintained by crossing with this control lines. Okay. So if we take the tissue from this and if you do the clonal propagation, all the lines will be fertile only. It is not sterile. So we can't use clonal propagation method for male sterile lines. Yes, option B is the right option. Next, which of the following enzyme finally transfer the ubiquitin to the target protein? Ubiquitin to the target protein. Which of the following enzyme? We know there will be three enzymes, E1, E2 and E3. This will be involved in what? Yes, 26S. You observe properly. 26S proteasome complex. These are enzymes. First itself we have to eliminate this one. Okay. First itself we have to eliminate that one. Uday Lakshmi. Okay. First itself we have to eliminate that one. It's a complex, proteasome complex. They are asking which of the following enzyme finally transfer the ubiquitin. Okay. You just observe here, students. There will be three enzymes. Okay. First is ubiquitin activating enzyme. Activating enzyme. Okay. Then first is ubiquitin conjugating enzyme. Next, activating enzyme. Next, ubiquitin transferase or ligase enzyme. Okay. So the third enzyme is the primary enzyme that will transfer the ubiquitin from ubiquitin to the target protein. This is very important thing. Okay. You just go through about proteolysis. Okay. How the proteins are degraded by 26S mediated proteasome. Okay. Yes, E3 is the right option. Yes. Yes, students. Yes. Next, which of the following condition will favor the synthesis of beta galactosidase in E. coli? Simple question. Simple question. This is simple. You can solve. I know. This we have seen in our class also. Which of the following will favor synthesis of beta galactosidase in E. coli? Under which condition it will do? Whether there should be excess glucose or excess lactose? Yes, excellent. There should be an excess lactose as an carbon source. Yes, very good students. Next, conventionally, observe this question properly. They are asking Tata bugs and minus 35 bugs are denoted in which strand of the DNA? Which strand of the DNA? Okay, for example, this is the double strand of DNA. For example, here promoter regions are there. Minus 10 and minus 35 regions are there. This should be present whether in coding region, whether in coding strand or template strand or sense strand. What will be the right answer, students? Transcription will always happen in at which stand? Template stand, yes. Any guesses? What will be the right option for this? The conventionally the promoter regions will be present in which stand? Whether it will be present in template stand of DNA or coding stand of DNA, sense stand of DNA, or on the same stand which is used as coding stand of DNA downstream to +1 site. Any guesses? Pakka, this will not come downstream to +1 side. This will not come. This we can easily eliminate. Yes, any guesses? It will be present in template stand. Any guesses? Okay, Uday Lakshmi is saying template stand. Any guesses? Yes, Tejaswini is saying coding stand. Mani is saying template stand. Template stand means what? Template stand means what? Template stand means the stand that is complementary to coding stand. For example, this is the coding stand. The stand that is complementary to template stand. So this is what? This is template stand. Okay. Where it will present? This will be transcript to mRNA. This mRNA will have similar structure with the coding stand of the DNA. So what will be the right option? Now you tell what will be the right option? Whether it will be present in template stand or coding stand. Template or coding? Now you can tell. Yes, you can see this, then you can tell. Yes. Any guesses? Just type your comments there. Any guesses? It will be present in coding stand only. Okay. It will present in coding stand. Yeah. It will present in coding stand. This RNA polymerase will go and bind here. That will initiate the transcription. Because of this, this template will be transcript to mRNA. This mRNA will be complemented to the coding stand. So the right option is what? Coding stand. Okay. Which of the following enzyme is used in second strand synthesis? They are asking second strand synthesis. We know mRNA will have 5 prime to 3 prime. Okay, this will have poly A tail will be there. For first strand synthesis, we will use reverse transcriptase, so cDNA will be synthesized. For second strand synthesis, what we will use? There are two different strategies are there. One is replacement method, another is self-priming. This I have discussed in our class itself. Okay. Replacement method and self-priming. Here what we will use? We will use RNase H enzyme. This we have already seen in our class. So, RNase H will be the right option. Okay. Which of the following formula may be used to estimate the concentration of RNA by UV spectrophotometer? Okay. We know 260 nanometer we will use for estimating the DNA or RNA content. 280 means what? That will be for protein content. If the ratio of 260 by 280 is 1.8 means what? 1.8 means there will be a 50 nanogram of DNA per microliter. This is very important concept. You have to just remember. If the ratio is 1.8, it will have 50 nanogram of DNA per microliter. If RNA means that will be there 40 nanogram per microliter. Excellent. Already one person has answered. So 40 into 260. Excellent. 40 into 260. Excellent. Next question. Large fragment of DNA is separated by large fragment of DNA Our chromosome is separated by which technique? Yes, this we have already seen. Pulse field gel electrophoresis can be used. Here, large fragment of DNA can be separated. Entire chromosome also can be separated. Okay, agarose and polyacrylate gel electrophoresis, there we can't resolve large fragments. Yes. Which of the following step of polymerase chain reaction operates at high temperature? They are saying which step will operates at high temperature. We know three steps will be there. First is what? Initial denaturation will be there. Okay. We will keep denaturation at around 95 degrees Celsius. So the double strand will come converted to single stranded molecule. Okay. Then we will keep at what? Annealing temperature. Annealing. Okay. Here our primer will go on to bind here. That will be based on the primers that will done. Third is what? Extension. We will keep at 72 degrees Celsius. So the right option is what? 95 degrees Celsius denaturation. Excellent. 95 degree desaturation. Next, which of the following DNA sequencing method uses dideoxynucleoside adenosine triphosphate? Which method will use dideoxyadenosine triphosphate? Which method will use this one? Whether Maxim Gilbert or Sanger or chemical method? Yes, Maxim Gilbert is also known as chemical method. Sanger is also known as what? Sanger is also known as what? Which method? Chain termination method. Chain termination method. The name itself it's there. This is not a dideoxy. This is not a deoxy. If deoxy ATP is there means it will lead to synthesis. Synthesis we can do. Dideoxy means it will inhibit, it will terminate the chain of extension. So the right option is what? Sanger sequencing. Clear? Sanger sequencing is the thing where they will use dideoxyadenosine triphosphate. Okay, so Sanger sequencing. A protein coding gene of an organism with unknown genome sequence can be isolated from. The question itself is there. Protein coding gene. That means what? cDNA library. Okay, protein coding gene means what? From cDNA library we can easily isolate from unknown sequence. Okay. This will not come. Gene specific primers. The name itself is there. That is an unknown sequence. How we can design a primer? So this will not come. Then degenerative primers, that also we can use. If any homologs means we can use genomic library that will have a introns that will not give exclusively the protein coding genes. So the right option is cDNA library. Okay. Expression of gene can be measured by this we already seen. RT-qPCR we can use, reverse transcriptase PCR we can use. Okay, RT-qPCR we can use. Apart from that, what? In RT-qPCR, what they will do? Reverse transcription they will do. So cDNA will be synthesis. This cDNA will be what? This will be counted. Okay. So based on CT value, this I have already told in the class itself. So the right option is what? Reverse transcribing the cDNA of the gene followed by polymerization will be the right option. Yes, B is the right option. Excellent. Next, which of the following statement is true? They have asked about RNA polymerase. RNA polymerase lacks nuclease activity. Yes, this is true. The error rate of RNA synthesis is much higher than DNA synthesis. Yes, these both are true. Yes, both A and B are true. Which is the true for 35S promoter? This is very simple question. 35S promoter. It's an what? It's a type of, yes, excellent. It's a type of constitutive promoter. It's a type of constitutive promoter. Yes, this is the right option. Which of the following scoreable marker? They are saying here scoreable marker, fluorescence, bright green color. Any guesses? Which of the following scoreable marker? Fluorescence, bright green color. Yes, green fluorescent protein. Excellent. Okay. This is antibiotic selection neomycin phosphotransferase. This will be what? This is luciferase, firefly luciferase. Based on luminance, they can select. Okay. So the right option is green fluorescent protein. The term heterosis. Heterosis means what? That was given by Shell. This is very simple and direct question. This you will solve, I know. Okay. Next, the type of chromosome in which centromere is located at the end of the chromosome. You have to understand here. Okay. The centromere is attaching at end of the chromosome. Like this is there means what? It's a telocentric. Metacentric means it will be there at middle. Meta. Submeta means at the tip. Okay, this is clear. So, yes, telomeric, excellent. So, telomeric will be the right option. Which of the following method of recombination in bacteria is DNA sensitive? This I have told in our genetic engineering class. It's a DNA sensitive method. Which will be the option? Which will be the option? Which will be the option? Hmm? Which will be the option? Yes, transformation is the correct option. Okay. Transformation means what? It's uptake of naked DNA. Okay, naked DNA by bacterium. Okay, so if DNA is present here, this may cleave this DNA. Okay, so the right option is what? Transformation. Transformation is DNA sensitive. Another thing, they are asking about in which technique antibodies are required, in which technique antibody is not required. We have already seen in these things in our technique in biochemistry class. Okay. Y2H, pakka they will not use antibody. They will use expression analysis they will do. Okay. BIFC, the name itself is there. It's a fluorescence based method. Okay. It's a fluorescence based method. Split ubiquitin assay. Here also same thing. Here they will not use antibody, but in pull down assay they will use the antibody. So the right option is what? Pull down assay. Okay, students. Next, the headquarters of ICR is located at. Simple and direct question. Headquarters of ICR is located at where? Hyderabad, Lucknow, New Delhi, Karnal. Where is the headquarters of ICR? See, 2025 they have asked who is the DG of ICR. 2024 they have asked where is the headquarters of ICR is there. You have to know about the ICR, TK, when it started, what is the motto of ICR, TK, how many institutes are there. These things they can ask in general agri, TK. So you have to focus that one, TK. What are the institute, where it is located? That is most important thing, TK. So ICR headquarters is there in New Delhi. Which of the following cell organelle has connection with male sterility? Plant male sterility. Which of the organelle is connected with male sterility? Any guesses, student? Yes, any guesses? Mitochondria, chloroplast, peroxisome, and endoplasmic reticulum. Yes, chloroplast. Any guesses? Mitochondrial male sterility. You just remember MMS. Okay. Mitochondria is the primary organelle. Okay. If some defect is happen, that will lead to male sterility. Okay. This is another important thing. Mitochondrial male sterility. Okay. This I know. This you all guys will answer. Which of the following is required for CRISPR-Cas approach of gene editing? Which is required? Guide RNA, microRNA, small RNA, long non-coding RNA. Which will require, students? Whether guide RNA is required or microRNA is required or small RNA or long coding RNA. Which will be required? We already seen 2025 they asked the same question. This is repeated question. So they are repeating the question also. Yes, guide RNA. Okay. Repeating the question. Guide RNA is the right option. Which of the following are application of proteomics? Several questions they have asked from technique in biochemistry. You have to be very strong in techniques. Yes. Which of the following is application of proteomics? Okay. Mining of protein in a sample, yes, we can do mining. Protein expression profiling, yes, we can do this protein expression profiling. Determination of native structure of a protein. How we can determine the native structure? We can't determine the native structure of proteins by application of proteomics. Okay, this can be done by what? Native page. This will give just a three-dimensional shape or what are the amino acid presence. Okay, mapping of protein modification. This also we can see. So A, B, and D will be the right option. Yes, yes, excellent. Ganesh, Mani, excellent, excellent. D will be the right option. This we have already discussed in our class. Different scientists along with their discovery. This we already seen. This is very simple question. You have to answer. Semi-conservative DNA replication. Who has given that one? Reverse transcriptase enzyme. Who discovered? Gene editing. Who got Nobel Prize in chemistry or medicine? In which field they have got a Nobel Prize? Insulin. Who has sequenced that insulin? Yes. Semi-conservative DNA replication. Watson and Crick. Yes, this we already know. So first will come 4. First will come 4. Okay. Reverse transcriptase. Temin and Baltimore. Yes, here it's there. Second will come 1. Gene editing. Yeah, Charpenter and Doudna. This we have seen in our class. So last option is this one. So option C will be the right option. Excellent. Next, in a monohybrid cross, see, continuously they are asking biotech question. You listen properly. We are going to solve this one. In monohybrid cross, which of the following is always completely heterozygous? Okay. Which following is always completely heterozygous? Okay. We know in monohybrid cross, F1 generation will always have an heterozygous. Okay. So complete heterozygous will be there in F1 generation. Yes, excellent. F1 generation. Second question. You have to observe this properly. Okay, this we are going to solve. What is the question? Tallness is a dominant. They are saying tallness. T is for tallness. It is a dominant to draftness. Draftness is small t. White, while red flower is color is dominant. Red is dominant and white is resistant. Clear from the question I am saying. Next they are saying the heterozygous condition result in pink color. So R means pink color will produce. Okay. Now they are asking if a plant is dwarf. If a plant is dwarf, okay. Dwarf red snapdragon is crossed with plant having homozygous for tallness and white flower. You see here a plant that is dwarf. Dwarf means TT will be there and red snapdragon. Red means which color it will come? It's a dominant. Correct. R will come. This is clear. If they cross this with homozygous tallness and white flower. Homozygous tallness and white flower means what it will come? White flower means RR. So if you see this gametes will come, this will come. So ultimately TR and T capital T small t capital R small r gametes will form. This will form. This is clear now. Okay. But what they are saying, what are the genotype and phenotype of the F1 individual they are asking. Okay. What will be the genotype and phenotype of F1 individual? From this cross we know these things are producing capital T small t, capital R small r is producing. Capital T will be dominant. If dominant means that will produce tall only. Yes, this is correct. Capital R small r, if capital R and small r is there means that will give pink color. Okay, capital R and small r is there means that will give pink color. So the right option is what? Option D. Tall pink, excellent. Tall and pink, excellent. Money. Money, excellent. Next question. The process in eukaryotic that result in production of gametes. Gamete production, this is simple question only. Meiosis, cell cycle. This is direct question. I know you will solve this one. Take meiosis. Next, the situation where each allele produce a protein. Each allele is producing a protein that can be detected in heterozygotes. It's called. See, there are two alleles are there. Allele 1 and allele 2. Each is producing a protein that can be detected in a heterozygote. So what will be the correct answer? If dominant is there means single thing will be produced. Okay. This may be the typical. Yes, excellent. Incomplete dominance will be the correct answer. Excellent. Udaya Lakshmi and Mani, excellent. Next question. Prions. Simple. Prions are what? Yes. Prions are proteinous infectious particle. Prions are what? Proteinous infectious particle. Excellent, students. Yes, we have completed the first part of 2024 previous year question paper discussion. Okay. Remaining things we will resume at 2:30. Okay. We will resume at 2:30. Any doubt is there, students? Any doubt is there? Kindly let me know any doubt is there. If no other doubts means we can complete the session now. Any doubts? Yes. Yes. No doubts means we are closing the session now. Thank you, students. Thank you.